So welcome to the stick facts case study. In this case study, we're going to be looking at a relatively simple design, treatment compared to control, but with a couple of interesting wrinkles around data borrowing and sort of slightly complicated dropout situation. I'll show you, the, the the background to the trial, some of the initial design ideas, actually, and what the design looks like entered into fact. And then I'll also finish by describing the iterations we went through with the sponsor when they'd seen the initial design. I think that iteration is something that is such an important part of simulation guided design, and it's perhaps I don't think it's fully appreciated. What can go on and how important it is by people who haven't actually done this. So the background to the trial first. So I'm not actually going to tell you what the actual indication, compound, or company are in this case. I'm anonymizing that, but I think everything will be sufficiently clear without you needing to know that. So the drug in question is a treatment for a chronic condition. It's being developed, it's shown efficacy, but a safety study needs to be run. And a safety study had been run, but something had gone wrong during the trial and it had to be abandoned. So this is a safe, fairly conventional setup where the safety study was showing non-inferiority to a placebo with a thing called the MACE endpoint. Looking at the data of this abandoned trial, the incomplete data was very, very promising. Indeed, the hazard ratio was less than 1. The FDA required the safety study to be rerun, but they were prepared to allow some of this promising data to be included in the new trial through some data borrowing. They accepted that this would inflate type 1 error, and they were prepared to accept 0.05 single-sided in the new trial. As in effect, as a consequence of doing the data borrowing, it was accepted that the design of the analysis would have to be Bayesian to do the borrowing, and that meant that a conventional on a group sequential thing wasn't going to be possible. That we'd need to do Bayesian throughout, which is one of the reasons the trial came to Berry Consultants to do. So this endpoint is time to event. It's called MACE-3. It can also be called hard MACE. And the endpoint is the time to either cardiovascular death, some non-fatal myocardial infarction, or a non-fatal stroke. And in the population that was going to be included in the trial, the expected probability of having one of these events in a year was 2%. And we can convert that to a weekly hazard rate, taking log of 1 minus 0.02 and dividing it by 52, negating it to give us our weekly hazard rate of 0.00039, which is what we're going to use in fact. Something that was also considered was expanded endpoint MACE-4, which includes the endpoint, also an event of being hospitalized for unstable angina. And the hope would be in that case that the increased event rate would lead to a smaller trial. Now, the data borrowing was done in a fairly straightforward manner through a simple power prior. So from the prior data, which I'm not going to give the details of, from the 90%, 95% confidence interval, with some lower and upper bound, this was converted to the log hazard ratio and matched with a normal with a normal distribution, which would have had some mean and some standard deviation. And if we use that as a power prior, and we're going to apply some weighting, in this case 0.25 to take just a quarter of the weight, we simply modify it by dividing the standard deviation by the weight factor. But it was also, particularly the FDA's opinion, that there was no expectation there would be any benefit of this treatment on the safety issue. So instead of using the observed favorable mean of X, it was set to 1, so just being no different from control. So in our primary analysis, we use the prior of log of 1 with a standard deviation of 0.29. So the effect of this dilution looks like this. The fully borrowed prior would have been this black curve, and the shifted 25% curve, so shifted, so its mean is 1, and then weakened to 0.25. This curve would have looked like that. So you can see, and there's a dashed line at 1.4. You can see how the prior from the borrowed data at full strength would have already given you full evidence that you were non-inferior. So that was also part of the reason for weakening it. An additional complication was dropouts. This was a little more complicated than normal. So there's a background dropout rate of 3% per annum, but there's a much higher discontinuation rate. 27% of patients discontinued in the first 0.3 year, and after that, 36% discontinued per annum. But a patient that discontinues is not an immediate dropout. They're still followed up for a year to see if any safety events occur. So a patient that discontinues in their first 0.3 of a year stays in the trial for a year, plus the 0.3 for which they were actually taking the treatment. So our dropout rate for a subject is based on a hazard rate derived from this 3% probability per annum for the first year, and then for weeks 52 to 67.6, that's 0.3 of a year. The hazard rate is 0.3 plus this 0.27 divided by 15.6. And then thereafter, the dropout rate is 0.03 plus this hazard rate due to this 36% per annum dropout rate. The rest is relatively straightforward. The allocation ratio is just going to be 1-to-1. The recruitment is expected to be on average 750 a month, but it'll take about 22 weeks to get up to that speed. That means that a trial size of 5,000 subjects would be recruited in about 42 weeks. We're going to use the standard FACS Bayesian time to event analysis, which is a piecewise model. We're going to use pieces that are a half year in length, so 0 to 26 weeks, 27 to 52 weeks, 52 weeks plus. And the prior for each piece is going to be set at the mean hazard rate that we expect for the control hazard with a weight of 1. So that corresponds to an expectation of a 2% probability of an event per annum. For non-inferiority, we're allowed a non-inferiority margin of 0.4 on the hazard rate scale. So what we're interested in is the probability that our hazard ratio on the treatment is less than 1.4. We're not interested in testing against the lower bounds. This isn't an equivalence test. We simply have to be less than the upper bound of the non-inferiority bounds. And sufficient evidence of non-inferiority would be the probability of the hazard ratio being less than 1.4 is greater than 0.975. And this is chosen through simulation to control the type 1 error at 0.05, given our prior informative prior. Now, there's already some ideas for making life more complicated. So we started by simulating this fixed trial, but then we thought, well, we know we can do better than a fixed trial. We can add interims and allow the trial to stop. So we looked at adding interims at 50% and 75% information with cautious boundaries that roughly maintain the type 1 error and power with the same maximum number of events, and then more aggressive boundaries with slightly larger number of events, 230. There was also an idea to, instead of using the full population that had been tested before, would be to enrich the population with folks more likely to experience a MACE event, and that would be typically type 2 diabetics or people with cardiovascular disease. There was also the requirement to evaluate the design using this MACE-4 endpoint. Can we be smaller, quicker, and cheaper? So we'll just look at entering this base design in facts so you can see what it looks like. So it's going to be facts core with a time to event endpoint. And then we have these various details that we require to enter: the maximum number of subjects, our maximum number of events, the follow-up time after full enrollment, which was 6 years. Facts that events indicate failure. We're going to recruit subjects continuously, as one normally does. And we just got the two treatment arms, control and treatment. For the virtual response, we want to explore 3 different possible rates on the control arm of 1% per annum, 2% per annum, and 3%, yielding these weekly hazard rates here. And we want to explore dose response profiles with hazard ratios of 0.8, so being actually superior, of 1 being equal to placebo and safety respects, and then 1.2, 1.4, 1.6, and 1.8. In fact, it turned out the 1.8 really wasn't very interesting. The success rate was about 0.1%. And so we quickly dropped it from subsequent simulations. 1.6 was pessimistic enough. And of course, in here, in sort of conventional terms, the hazard ratio of 1.4 is our null hypothesis, and a hazard ratio of 1 is the alternate. So we'll be majoring on looking at the results for those 2 scenarios when we look at the simulation results. We have the execution parameters, 173 a week, ramping up to 22. And then these dropouts where we've got these 3 different rates for dropout over time. Then we'll be creating our quantity of interest and the decision quantity. We set up the hazard model for the control, the dose response model, simple one-to-one allocation, and then a simple success futility rule. Get rid of some of this. There's a new core time-to-event design. At this point, we're just doing the base one, so there's no adaptive features. Our sample size is 5,000. Maximum number of events is 212. Our follow-up is 312. events indicate failure, and we're going to recruit subjects continuously. There are no visits. Events are deemed to be observable whenever they occur, not restricted to some visit. We have a control arm, and we have one other arm, which is our treatment. We're not going to do any variants at this point, but that's what we used when we were exploring the sample sizes and settling on 5,000 in the first instance. And then we have the virtual subject response, which in time to event is done in two parts. One is specifying the hazard rates for control, and the other is specifying the dose response of the treatment arms were multiple. So as I said, we had three.
Sorry, will you flip
Yeah,
back to the study info tab?
sure.
You have 500 as the maximum number of subjects there.
I missed. Thank you for spotting that. Thank you. So, well, I'll use 1, 2, and 3 here because they correspond to my 1%, 2%, and 3%. So we're just going to have a constant hazard rate over all time, and that's going to be 0.000291 for 1%, 0.000389 for 2%, and 0.000487. Now, in hindsight, I think actually these are probably, this degree of uncertainty is probably a little bit extreme. That just represents kind of, 100% uncertainty on the low side and 50% uncertainty on the high side. Maybe 1.5 and 2.5 would have been sufficient spread rather than 1 and 3.
Will you flip through your control profiles real quick?
Yeah, what happened to that one? Did I not? Oh, I hit enter or something. Here we go. Thank you for spotting. Did you spot that? Thank you.
Yeah, we have a very detailed observer.
Excellent. And then we create some simple dose response profiles. One, 1.2, 1.3. You have to be a little bit careful what labels you put in these because these get used in file names. You can't use characters in there that will upset Windows file naming. In fact, it attempts to spot all those for you now. There were times when it didn't. Right, and we'll just check all those. Good. So the accrual was, as we said, 750 a month, which equates to 173 per week. We don't start off at that rate. We say, no, it takes about 22 weeks for us to reach that, but we just assume that the accrual rate will ramp up linearly. And that gives us, on average, about 42 weeks of recruitment. Dropouts are more complicated. We have to do them in these 3 segments. So the first dropout rate is for the first 52 weeks. Then there's 52 to 67.6, and then 67.6 onwards. And in the first Get my notes here. And the first period, well, we want to do it by rate. The dropout rate is 0.123586. In the second period, the dropout rate is 0.02076. That's the folks that discontinue in the first third, but are then followed up for a year. And then the folks that are discontinue after that, we model with a dropout rate of 0.00917, which gives us this net rate of about 30%. The quantities of interest, we can actually get rid of some of these. And what we do is we say it's a non-inferiority study, and the non-inferiority margin is 0.4. And that creates for us here the posterior probability test that we want. And then we create a decision based on that, decision quantity of interest based on that, the probability of this for the treatment arm. Now, yeah, maybe we could make facts a bit smarter and notice we've only got one arm and populate that automatically. But then on the design, we're going to analyze the control. We set up separately how the control arm is as a rate is analyzed from the dose response of all the treatment arms. So here we're going to specify 3 time segments, 26-week intervals. And for each one, we put in the mean rate, the same as the 2%, the median rate we're going to simulate on control. But in all cases, we only give them a weight of 1. So these are just kind of centered in the right place, but they're very uninformative. And the dose response, we use a simple independent dose model, and we simply use this smaller standard deviation, making it informative and centered on the around 1. Hazard ratio of 1. We're not going to do any frequentist analysis, simple one-to-one allocation. And then the last thing remain to do is to set up the success and futility criteria, which is this posterior probability has to be greater than 0.975 for success. So if it's less than 0.975, that's going to be failure. And now we have our various scenarios. So you can see we've got quite a number of scenarios. We've got the 3 control hazard rates, and then we've got the 0.8, 1, 1.2, 1.4, 1.6, the 5 different hazard ratios, and each one crossed with a different control hazard rate. Now, this is actually quite a slow trial to simulate in facts terms, because we've got partly because it's simulating time to event, and partly because we've got such a large sample size. So I've only done 100 simulations, but that comes through nicely in a few seconds. So we can see we've got fine power if the hazard ratio is indeed less than 1, 0.8. If it's 1, we can see we have a power of around about 0.8, which is our target. and around the null 1.4, we've got some varying results. 0.06, 0.08 is a bit high, and 0.09, which is also high. But this is a very small number of sims, so we can't be too worried about that yet. We can do some other things like check how much we're observing, so we can see the mean. We are indeed able to observe 2-on-2 events in most cases, but there are some where we don't actually get to see 2-on-2 events, and this is because of the dropout. You can see if the underlying control rate is low, and of course then the treatment rate is also low, then we're not seeing the full 2-on-2 events. And also where the hazard ratio is below 1, then also increases the likelihood that we don't get to the full 2-on-2 events. And then we can also see the mean duration. And some of these go way out, and this is partly because of the dropout. So where the actual towards the end, we haven't seen enough events. Almost all the population has dropped out, and we're following just a few tail enders in the desperate hope that we'll get a couple more events. And sometimes we'll actually get to the end of our 5-on-2 follow-up, or the last patient will finally drop out and we've not seen 2-on-2. But you can see there's some scope for this being really quite a long trial. So I hope that showed how easy that was to set up in FACS, and how relatively quick the simulation results were. And doing 10,000 simulations just took a little bit less than 10 minutes. And I've just picked out the kind of 3 key operating characteristics: the probability of success, the probability of failure, and the mean duration. And here you can see in the low control hazard rate, we're going all the way out to over 300 weeks. The power is a bit lower than 0.8, and the type 1 error is about 0.06. For our median case, our power is 80%, and our type 1 error is just below 0.05. And only if we were lucky enough to have a treatment that was more safe than placebo would we have some risk of running out to quite a long trial. Otherwise, the duration is going to be kind of 5 years or less. So to look at the adaptive trial, we've put that little bit of effort in to create our fixed design. So we save a copy of that, we make it adaptive, and we boost the maximum number of events a little bit to compensate for early stopping for success, and make sure we output cohorts files for all these guys, so we can start to look at the results over all the cohorts for what the stopping boundaries could be. So I've got this in no early stopping, but with interims. And in order to look at the stopping boundaries, I'm just going to restrict things to the median, the 2% hazard rate, and the alternate hazard ratio of 1 scenario, and the null hazard ratio of 1.4 scenarios. Even though I'm not going to look at a single, I'm going to use the across scenarios graphs, and I'm going to use the, oh yeah, just to remind you, I set up to simulate 1,000 weeks So output the weeks files, the things that record every interim for each one of the 1,000 simulations we've got here. So then I can do this interim versus final result scatter plot. And here, what I can do is I can use the graph to reinterpret the results. And the scatter plot is showing me on the x-axis what the posterior probability of being below 1.4 was at the interim, and on the y-axis we've got what the probability of being below a hazard ratio of 1.4 was at the final analysis. And then the color of the dots indicate the outcome. So if they're red, they're futile at the end. If they're green, they're successes at the end. The successes are all up here at the top, because to be a success at the final analysis, the posterior probability has to be better than 0.975. If they were, so there's a couple of default thresholds for stopping here, which have been put in, that stop early for success at 0.9 and stop early for futility at 0.2, and it's evaluated at the first interim. So these aren't very good thresholds, as we can see, because if we look at the futile trials, look along the interim value, we could go all the way out to 0.6, and only then would we encounter the first trial that was eventually successful. So we could increase the utility boundary to 0.6. But we've also got the opposite problem here, because this interim value is way too low, that we've got all these pink dots, which represent trials that had a interim value of less than the success boundary entered here, 0.9, but were then futile at the end. So they were less than 0.975, which is why 0.9 was not a good default guess in this instance. So if we boost that to 0.99, say, and I've adjusted the futility criteria too, we can get it to redraw. Now, because I've just got the two scenarios picked and there's only 1,000 simulations of each, the redraw time is quite nice and quick. If you wanted to use this method and you had several scenarios and maybe 10,000 simulations and I put 10,000 weak files, you had to be much more patient about the redraw. It'll take quite a lot longer. But you can see now that we've got quite a decent probability of stopping for futility. We've got quite a decent probability of stopping for success. There's a tiny, we can see there's a few incorrect stoppings for success, and we can dive in to look at that in a bit more detail by modifying the axis range. So at the interim values, probably don't want anything below 0.975, and then for the final values, we want about 0.9. If we zoom into this upper corner, you can see that with a stopping bound of 0.99, we have all these light green spots, the things we stopped early for success that would have gone on to be final successes. And then we've got these few pink dots where we've stopped for successes, things that would have gone on to be futile. So this is quite likely to be our type 1 error inflation. So I actually picked 0.995 to reduce this error inflation, and then we can also look at how that looks on the second interim. So 0.995 on the second interim is also nice and cautious. And if we put the axes back, we can see at the second interim, we can shift the utility boundary a little bit further to the right and still not make any type 2 errors. Maybe we can put that at, say, 0.7. Go. And so with the second interim, we now have almost a 50% chance of stopping early for futility. I've just got those captured in slides. So we stop early for success at either interim if the posterior probability that the hazard ratio is less than 1.4 is greater than 0.995. We stop for futility at the first interim if this posterior probability is less than 0.6, and we stop early for futility at the second interim if this probability is less than 0.7. I ran these simulations earlier today, and I was horrified that despite having lots of early stopping, it would made hardly any difference to the duration of the trials, which didn't make any sense at all. And I realized what I'd done was failed to set that I don't want to follow up after stopping for success. So on the tab where we've set up the interims, there's a checkbox here which is checked by default, because in the continuous and the dichotomous setting, if you stop for success, you may well want to follow up, complete the patients, and check against your final success criteria. In time to event, particularly if follow-up is 6 years, you don't want to do that. So that needed to be unchecked. And then this is the simulation now with the stopping boundaries entered. So 0.6, 0.995, 0.7, 0.995. I found that this did cause some type 1 inflation, despite having been tried to be a little bit careful with or cautious with these stopping boundaries. And I had to set the final test threshold to 0.98. So what I did was we picked the hazard, we picked a sort of median null. So the null case with 1.4 and the 2% probability of an event on the control arm. And then we use the explore final success criteria and said what we put in a target rate, which is actually the default of 0.05. And then it tells us at the bottom what our threshold would have to be at the final success criteria to keep success below 0.05. In this case, it's 0.9796. So that's 0.98 in normal money. Having increased the final threshold to 0.98, of course, that meant we lost a little bit of power. So the total number of events was increased marginally from 212 to 230. And that gave the final array of results. And then we can look at that in the table. So this is the orange represents the adaptive results, and the blue for comparison, the original fixed results. And here we, as well as looking at the probability of success and the total probability of success and the total probability of failure, we look at also what proportion stop early for success or stop early for futility. And we can see in our sort of scenarios of primary concern, the median control rate, the hazard ratio of 1, our alternate, we've got a 50% chance of stopping early. We've got an overall success rate of 0.8, and our duration has come down from 200 weeks to 174. So on average, we'd expect to save half a year. And under the null, again, we've got over 50% chance of stopping early for futility. The overall success rate is just below 0.05, and the duration is 124. So we've taken 20 weeks off compared to the fixed duration. So just in terms of time, the um, the adaptive solution looked pretty promising, and certainly for some of the if we were unlucky with the control rate, then this was offered some quite significant savings, particularly if the treatment was very safe.
I have a question, Tom, if I could interrupt.
Yes. Oh, yeah.
Once the simulations are finished, the design's finalized, and you moved on to the real trial, would you run these analyses through the analysis tab in Fax, or would you use another program like SAS or R to do that?
I think for this we would, because it's a Bayesian analysis, we would use facts, and that has we have done that on a few occasions. Quite often, the actual trial has got some little additional wrinkle in. So the simulations and the design has been done using facts, but then they want to add something like a covariate analysis, which facts doesn't support, and that requires the analysis to be written and then done using R, and if it's a Bayesian, sort of R and Stan.
Yeah, makes sense. And when you were moving all those futility numbers around, were you considering them binding in the sense that you felt that you could buy back type 1 error, and maybe you explain what that means a little bit in your response, or are
Uh,
they non-binding?
yeah, that's a good point. So in the fact simulations, if you set up stopping for futility rule, then the effects will be binding. And I've only used simulation results where the effects were binding. So yes, these numbers are all assuming a binding futility rule. And what that means is that um, we guarantee or we pledge to the regulators that we will stop at the interim if the futility rule is met. We won't change our mind and we'll carry on anyway. And of course, the regulators have said in their guidance on adaptive designs that if you do that, you have a futility rule and use a binding, assume that in its type 1 error control, because stopping for futility will reduce your type 1 error very, very slightly. Then they will expect you to hold to it during the trial. If you didn't want to use a binding futility rule, you'd need to do separate simulations where you only stop for success without the futility rule in, and then adjust everything to show that under that setting, you still controlled type 1 error. And then having set up your success criteria in your final analysis to show that type 1 error was controlled, you could then add the futility rules in and ignore the fact that you've now lowered the type 1 error a little bit further. But that would say success criteria do not assume that we necessarily stop at the interim for futility, and that would allow in the actual trial, if you're at an interim and you don't stop for futility, to override the futility stopping and carry on. So if you're doing binding for stopping for futility, it is well worth making sure your futility rules are pretty conservative, and there won't be any pushback to try to carry on the trial. And it will be so obvious that the data is not good enough that
Yeah, I
you
was
should
going to
stop.
say, since you were pretty aggressive in trying to draw your futility lines as close to the point where you stop observing final successes as possible, it makes sense that if you were
So
to simulate as binding, but then have the rule be non-binding, you'd probably actually have trials that would have ended up being successes if they had not stopped. So, um,
But yes, so if we wanted to be binding, we might make these rules more conservative. We might, instead of 0.7, we might use something like 0.5, something like that. So
Yeah.
it was quite clear that it just doesn't happen in simulation. And we test it with more simulations than 1,000, that that you're just not ever going to go to a successful position. And of course, at this point, what a posterior probability of being less than 0.4 of 50% is, of a half, that means actually your point estimate is going to lie pretty much on the 1.4 boundary. So I think people would say, yeah, it's pretty clear we're not going to be non-inferior. I think that would be
Yeah.
a relatively
We have a
safe
couple more
timeline.
questions. There's a related
Yeah,
one, so I'm just going to merge
yeah,
straight into it.
okay.
From like the output you get in facts and the visualizations you can see, is there a way to optimize the futility to success or success to futility flip-flop rates so that maybe you're only satisfied if that number is less than 3% or 5%? You could choose the thresholds for decision making to satisfy that without
Yes,
the guess and check method, sort of set a rule, simulate, and see if it worked.
certainly. But you can't. Facts currently doesn't have support for that, but certainly you could take all these individually simulated files. You can aggregate them. So uh, every simulation and all its interims are all in one great big CSV file. Load them into R, and then derive, just analyze the values of the quantity of interest of the interims in R, and say, if I want to say 1% chance of a flip flop, what would my cutoff be? Of course, to get the resulting cutoff value with much accuracy, you need to be using an awful lot of simulations to do that. But it can at least get you in the right ballpark quickly.
Yeah, and importantly, the rates specified in facts, in your example, I'm not going to do success to futility because you don't follow up, but you have futility to success stopping, or sorry, a futility to success flip-flop would be a simulation that stopped for futility, but after some amount of follow-up may have met the primary analysis.
Mm.
That's not actually maybe the most important quantity for this decision making. What you really want is what proportion of trials did I stop for futility that would have gone on and at any point later in the trial would have been successful. And so
Mm.
if you actually have a futility rule specified in your facts file, you don't get to actually observe what happens later. So generally, we would do this without specifying any decision rule. So every trial goes to the maximum, and then we would slice and dice with R, doing, pulling in this to R and say, what if I had set this futility rule? What proportion of the trials that I stopped for futility would have ever gone on to be successes is really an important quantity in that decision.
Yeah, yeah.
Yeah. Okay, and then there's a fun one here, and I don't know how you feel about doing it live. Could you demonstrate how to generate a simulation report using design report? I don't know whether you're using the dev version of Facts or where you are on that. So
And we just click design report.
you're using
I've
7.0?
Yeah,
Yeah,
I've
okay.
got under my options, I've got my R configured to pick up our studio. So it's automatically launched our studio, which was actually already running. In our studio, we can't run the code automatically. So what it does is it provides you with a file that you cut and paste.
You could also just click the source button in the top right.
Yeah,
And I will say, while
um,
this is calculating that
I think
in
we could.
7.1, soon to be released, we've streamlined this process a little bit, and it should, when you click generate design report, just open the design report. So it'll do all the R calculations in the background. And when you click design report, a Word document should just pop up for you. So that'll be a nicer way of doing this as that releases.
Oh, have I actually got to run something now?
Yeah, Tom, I
Didn't
think you have a
see.
mixed version. I think you have a blended version of facts. So I think the file you're showing is the 7.1 generate file, but the method you're using is the 7.0 method, which is weird. So you
Yeah.
don't need to run the concatenate commands. You need to run the code that's actually in those. So hit source in the top right.
Right.
Uh, okay,
So go in this
maybe
top
let's
right.
start over. Will you
Oh,
close
stop.
RStudio completely? Close the R profile and then close RStudio. Don't save. And now try again.
And make for the facts we wanted. Oh,
At the
now we've got
bottom.
something.
Now that's the
Yeah.
command you want.
Yep.
Yeah. And again, we've improved this process. So now you should have a report.
In the results folder. I'm not seeing the output.
Make sure your file paths you are in the same fax document, that folder you're opening and all that.
No, I'm not seeing. I'm not seeing the .doc file.
Go to fax then to make sure you're in the right
Sorry.
spot. Go to your fax file that you generated the report from. And then right click on one of those and open um, results folder. Yeah. Demo results. That's not where you're looking.
Ah, I thought I was in it. That's weird. I thought I was in it, but I did it from that one. Okay.
Yeah, it's the wrong file.
There
There you go.
we go. Okay. So for some reason
Easy.
it's
First try.
started having different faxes open has confused things. Oh, where's demo results gone? I don't have Word on in my Windows environment, which is why I'm just moving that. And there is demo report. So this is indeed analysis of the demo event and not the adaptive one. So there's a problem here that it's not done a very good job of the control hazard ratio. It's and again, we've got certain numbers that are smaller than it's expecting to output. And then the reported operating characteristics will be laid out. There's your design report. Never submit a design report that's been automatically generated without checking it first. It's very much intended to be a draft. Okay. So I won't be looking at the other options, but there was we repeated the design with modifications for type 2 diabetes and cardiovascular, and also a modified design with looking at using a MACE-4 endpoint. Things were adjusted both for the prior, the expected event rates, and of course the accrual rate. So the first simulation report looks to have taken about 2 weeks, judging by the time stamps on the files, which included a fixed trial, 2 versions with interims, a version with the enriched population, and a version using the MACE-4 endpoint. There's another report a week later, which concentrates on the enriched population, looking at smaller sample sizes, higher control event rates, a single interim at 50%, and 2 interims, one at 33 and one at 66%. And then also looking at the smaller sample size of 3,500 with a power of 80 and a sample size of 5,000 and a power of 90%. 7 weeks later, we got a revised protocol and we were asked to update it with the information from the samples. And we included a table like this, which doesn't explore all the different scenarios, but provides some additional interesting information, such as the probability on each arm of having a MACE event, the probability on each arm of being censored, the probability of success of the design, and also the probability of success if we didn't do any borrowing, if we had an uninformative prior. You can see the effect of borrowing there, increasing the power by over 10%. The probability of declaring superiority rather than just non-inferiority. And then in the worst case, for any set of simulations, what did we observe and still declare the trial successful in terms of the number of events on the alternate arm, on the treatment arm, the observed hazard ratio, and the adjusted Bayesian hazard ratio with the prior information. So would allow regulators to get a feel for whether all this borrowing and so on was going to give them a problem when they came to be given data to approve. Then it was noticed that actually dropout was even more complicated. That among those who discontinued, their actual background dropout rate was then 10%, not the 3% rate those who hadn't discontinued. So we put this in, we simulated this dropout for 1 million subjects, and then analyzed what that meant in terms of hazard rate. It meant this increased dropout meant that the most conservative control rate under the enriched patient population, the planned number of events weren't being achieved. And so the question back to the sponsors, do we just drop that scenario? Is it too conservative? Or are we going to have to increase the sample size? There was also a set of simulations run for a reduced band of non-inferiority. This seems to have taken about 3 or 4 days. We then looked at possibly shortening the follow-up, try to get the trial finished sooner, but allowing different sample sizes in terms of events. By now, we're just simulating the scenarios with a hazard ratio of 1 and 1.4, because these are the ones we were always looking at first. These weren't good enough, so we looked at increasing the maximum a little bit further to 225 events. And all told, that took about another week. Then everything went quiet for 6 months. And then finally, a revised protocol turned up, and we had to produce a report saying the various options that had been considered and rejected. And that took a further week. So these are some very common traits of simulation. There's this back and forth, which can be quite intense for a period, and then go quiet for long periods where other things have to be discussed, not with us. And these back and forth, they can be external reasons, like realizing the dropout was different, or they could be the sponsor shifting their requirements as a response of seeing the simulation results. Like, oh, well, let's use the enriched population, and can we make the trial shorter by shrinking the follow-up time? And towards the end, we also ran some type 1 simulations out to 100,000 simulations to prove the time on error control. And of course, the huge advantage of FACS in doing all this was the simple speed of getting the simulation results, and also the ease with which it was to make small adjustments to a design and resimulate. So this webinar series will be uploaded to our YouTube channel in December, and you might see them re-advertised as we write a sort of a blog, bringing out the kind of key points of each one. And for those of you who don't already know, FACS is free for academic and government institutions, and there's also a free several-month evaluation for commercial organizations. And then lastly, if you're not already, and you want to be on a webinar mailing list for our other subsequent webinars, just email me at tom@barryconsultants.com. And then lastly, for those of you who are already FACS licensees, thank you very much. And if you've got any training needs for 2025, please get in touch with me in December or January. Thank you very much. Any last questions, Nick?
Nope.
No. Great. Okay. Well, we'll wrap it up there, and there will be a webinar, a regular FACS webinar in December, where I'll be looking at machine learning to optimize things like stopping boundaries.
Great. Thanks, Tom.
Thank you.